Fundamentals of population genetics applied to organic heterogeneous material development and use. So the fundamentals, the most basic concepts and approaches. Well, as I told you this morning, there are two important things. As I always repeat during the presentations, all the questions, please go through the chat. We will try to answer as many as possible. And even in the case that some question is not answered today, we will revise the list of questions and we will provide to all of you our answers. But please, if I do not ask directly to some of you, please, any question through the chat. And for those who are interested in certificates or attendance in this unit, I will evaluate the quiz, a fast quiz at the end of the presentation, and also quite an easy homework based on the practical presentation I will show you more or less at the end of this unit. This both quiz and homework, please send to me and for double checking to Ana Maria Chorich. Well, and going further, what kind of things or approaches will be treated, we will see today in this unit 3, the fundamentals of population genetics. We are going to see the basic info and the parameters on the genetic structure of population, the most simple parameters. We will also see how the populations evolve with reproductive systems, the two extreme reproductive systems, autogamous or self-pollinated species and the allogamous or cross-pollinated species in a strict sense. There are, for your info, there are other intermediate situations, okay? But as I told you, I will simplify very much. The intermediate situations are treated with some parameters, with some coefficients of autogamy or allogamy to correct the formula used in autogamous or allogamous strict crops. But in this unit, I will show you what happens in strict autogamous crops or strictly allogamous crops. Well, in third place, we will go to have a fast review on microevolutionary forces. And later, I will provide you a practical example. I will practice with you with the PopGen server from Radford University. I will show you how this server works to reproduce, to simulate cases of population genetics and the evolution of the genetic structure of populations, depending on different macroevolutionary forces. They will take about 20-30 minutes because this is very important for you to understand, because the homework I will ask for you will be based on practicing with PopGen at home, okay? And after this practical example, we will finish with the quiz. I will give you 10 minutes for filling the quiz, 3-4 easy questions. And finally, the grow up part, take home message, and the homework. In concrete, what I'm hoping from you with the homework related to this PopGen server. Well, let's start with the first point, the basis of the genetic structure, to understand the genetic structure of a population, or what means genetic structure within a population, which kind of parameters work with that. Just one question. I'm listening to some noise, so please check that your phones and your micros are closed, please. Well, basics of the genetic structure of a population. We are going to be very basic. Again, we are going to work with just one gene, let's call it gene A, and two alleles, like the samples of problems mathematical problems with Mendel, okay, Mendelian problems. Allele A1 or allele A2. Well, considering that as many species we are studying, assessing the genetic structure of a diploid species, the different genotypes we can find in this population are only three possibilities, three combination in pairs of these alleles. So we have A1 homozygous genotypes, A1, A2 homozygous, A2, and the heterozygotic genotype, A1, A2. Okay? To follow in a very easy way, I have represented an ideal population made of these three genotypes, and each point represents one individual. So you have several red points which represent A1 homozygous individuals, A1, A2, the yellow squares, and A2, the plus symbol in green. So with this, you can count at home the number of individuals on each category or genotype. So this is the first important thing. So please, I will give you 1 minute to count the number of individuals 1, number of individuals 1, 2, and number of individuals which correspond to the genotype 2, 2, and also the total number of individuals in this ideal population. So I give you 1 minute to count the number of individuals. So please go. So, okay, the time is gone. Somebody can tell me how many individuals on each class, each genotypic class we have, and the total number of individuals?
Hello, everyone.
Hello.
I guess number N11 are 17.
Good.
N12, the heterozygous, 22.
Good.
And N22, 19.
And total number of individuals?
58.
58. Okay. Thank you, Roberto. Amazing performance. Good. So we have 17 individuals of the genotype 1, 22, 1, and 19 from the 2, homozygous. Okay, and a total of individuals of 58. Good. With this information, we are going to estimate the first simple parameters to define the genetic structure of this population. The first one are the genotype frequencies, which means the proportion or the ratio of each genotype on the total population. And it is so simple that dividing the number of individuals of each genotype to the total number of individuals of the population. So the frequency of individuals or genotypes 1 are equal to 17 divided by the total number of individuals, 58. The frequency of 1, 2, individuals 1, 2 are 22, which are the individuals 2, 2 divided by 58. And finally, the frequency of individuals 2, 2 are 19 divided by the total individuals of the population, 58. So please calculate on your homes. 0.293 is the frequency of genotypes 1. 0.379 is the frequency of the heterozygous individuals, and 0.328 is the frequency of the 2 homozygous. And if we made well the estimation, all the three should make 1, because the total proportion of the population is 1. So we have estimated the genotype frequencies. This is one of the parameters which define the genetic structure of a population. And the second one are the allele or gene frequencies. Okay, I have estimated the frequencies of the genotypes. Now it's time to estimate the frequencies of the alleles or genes. I have two alleles in the population combined in the three different genotypes, but I need to account the total number of alleles, 1, and total numbers of alleles, 2, in the population, and estimate the ratio or the frequency in the total population. So we have two ways to estimate. For instance, in allele A1, the frequency of allele E1 can be estimated as on the basis of the number of individuals. For instance, individuals 1, 1, you must consider that each of them provide two alleles 1 to the population, isn't it? So the number of alleles, total number of alleles 1, type 1, provided by these individuals is 2 by number of individuals 1, 1. Okay? Plus the alleles coming from other genotypes that without being homozygous for the allele 1, being heterozygous, they still provide allele 1, which is together the individuals 1, 1 multiplied by 2. Okay? So in the upper part of the ratio, I have estimated the total number of alleles A1 on the population. Double, twofold provided by each individual 1, 1, and 1 per individual 1, 2. Okay? This total amount of alleles A1 are divided by the total amount of alleles in the population, which is as simple as which is the number of individuals I have in the total population and the total number of individuals multiplied by 2, because each individual provides 2 alleles to the population. In this way, I could estimate the frequency of allele A1 as 2 by number of individual 1, 1, 17, plus only multiplied by 1, the number of individuals 1, 2, which is 12. And then everything divided by 2 by 58. So this makes 34 plus 12 above and 116 below, 46 divided by 116, which makes approximately 0.4. Okay. There is also another way to estimate this, the frequency of this allele, which is on the basis of the genotype frequencies. The frequency of the allele A1 is equal to the frequency of the homozygotic, which contains the allele A1, frequency of individuals A1, A1, plus half the frequency of the heterozygous, which contains this allele. If you make the calculation, you will see that it will give also 0.4. Okay. And also in population genetics, the frequency of this allele A1 is called with the lowercase p. This is very usual in population genetics. Okay. And in the same way, to estimate the frequency of allele 2, you need to perform the same calculations. You will estimate that the value frequency of allele A2, or call it Q, is 0.6. You did well the estimation. The sum of both should be also 1, the totality of the frequencies. Okay, so now we have defined well the genetic structure of this population on the basis of the frequencies of the genotypes involved in our study and the frequencies of the alleles. And what happened at the third level, the frequencies of the phenotype? Well, it depends on the interaction between the alleles. For instance, if we are in the case of the Mendelian situation where A1 dominates over A2, this is very easy. The phenotype of the A1 individuals will be the same than the phenotype of the heterozygous. So the total frequency of the phenotype A1 will be the sum of the frequency of genotypes A1A1 plus the frequency of the genotypes A1A2, while the frequency of phenotypes A2 will be just the frequency of the homozygous genotype A2A2. Okay. Alternatively, if we're in a case of codominance, partial dominance, intermediate inheritance, etc., I mean, any case where there is not a clear dominance between A1 or E2, that means that the heterozygous genotypes show a specific phenotype. So each genotype has one phenotype, and the three phenotypic frequencies are the same than the genotypic frequencies. Okay, so we can face two different levels, or two different ways to estimate phenotypic frequencies depending on the relationship between the alleles, dominance or other partial dominance or intermediate situations. Well, this is the basics. Okay, I will only show a bit complicated case just for your info and to keep here, because in real life, the traits are not all Mendelian, are not only based on one gene and or two alleles. For instance, we have also many cases where the gene depends not only on two alleles, A1, A2, but also on a third allele or even more alleles, okay, A4, A3, etc. But I will stay with three alleles just to show you, to explain you how can be this phasing, how multi-allelism can be phased to define the genetic structure of a population where more than two alleles are present in the population. It's quite simple. It's so simple like counting the number of individuals corresponding to each of the genotypes. In these cases, if we have three alleles, the different combination in pairs are six. A1A1, A1A1, individuals A1A2, A1A3, A2A3, or A3. Okay? So once I account, the different number of the different individuals, then I estimate their corresponding genotypic frequencies. Okay? It's so systematic. And what about the allele gene frequencies? Again, be systematic. For FA1, the frequency of A1, I would estimate the number of individuals who are in homozygosis carrying this allele. Number of individuals, 1, 1, by 2. They count twofold, plus the number of heterozygous 1, 2, plus the number of heterozygous 1, 3. And this total number of alleles coming from different genotypes which carry the allele 1, then it is divided by the total number of alleles in the population. So the total number of individuals by 2. Okay? Also, the other way to estimate the allele 1 frequency is developed below. And again, we give the lowercase letter p for this allele, for the frequency of this allele. And in the same way for allele 2, with the q as a symbol, as letter to represent its frequency. And in the case of allele 3, this n. Similar way to estimate. And in the literature related to population genetics, this third allele, the frequency of this third allele, is usually represented by the lowercase letter r. Okay, but just for your info, again, I will be simple. I will simplify and summarize the situation, because after this fast review about the main parameters to understand the genetic structure of populations, now we go to the second point, how the genetic structure of the populations evolve merely with the reproductive system, if we only consider the reproductive system. And as I told you at the beginning in the index, I will show the two extreme cases, autogamous species or allogamous species. So in the case of autogamous species, which are the ones who are self-fed generation after generation, and being simple, thinking just in order to understand what happens, thinking just in one gene and two alleles, imagine that we are in the most extreme case of this autogamous population. The individuals are only heterozygous. Imagine, for instance, that we are working with the F1 piece from Mendel. In the F1 of Mendel, only heterozygous individuals were present. So they represent the totality of the population. You can see in the F1 represented on the left that represent the totality, 1, while the homozygous, uppercase A, are 0, and lowercase a, the other homozygous, are 0, are not present in the population. Well, as many of you know, when I cross by themselves one individual, the segregation is quarter for each homozygous, and the heterozygous decreases to a half. So you can see that in the F2, we have 0.5 of uppercase homozygous and 0.5 lowercase homozygous. And the heterozygous individuals have decreased in the population to a half. And what happens in the next generation? In the next generation, the heterozygous, as it is self-fed, again, it decreases to a half, 0.5, while the homozygous, coming from the F2 as homozygous, they give again homozygous, but they are increasing thanks to the homozygous coming by self in the heterozygous individuals. So you can see that the F3, now the homozygous 375 each, and so on and generation. And what happens? After, for instance, 11 selfing generations, the heterozygous individuals have almost disappeared, while the homozygous encompass the totality, uppercase, half, lowercase. So this is what happened with strictly autogamous species, even coming from heterozygous individuals. At the end, they give a mixture of inbred lines or in highly homozygous individuals, 2 genotypes per gene. In the case of the gene A, we have individuals uppercase A and lowercase a. But the heterozygous almost disappear, or they are very rare, just in case that some cross-pollination may happen during the process. Okay, so widening this to other genes and considering the individuals, they carry genes A, genes B, C, D, etc. Here you can find represented what you can find, a mixture of individuals which combine different genotypes or gene combination, but highly homozygous, okay? With uppercase or lowercase alleles, but a mixture of highly inbred individuals, highly homozygous individuals. The corresponding part of the heterozygous in the genes has almost disappeared. And this happens on highly autogamous species. And what happens with allogamous species, the other extreme, with strictly allogamous species, the reproductive system in a strict sense, provides that all the individuals have the same opportunity to cross each other. So it's panmixia, it's highly cross-pollination, okay? And they follow the Hardy-Weinberg law in this, let's say, ideal situation, okay? On an ideal situation where the population is made of an infinite number of individuals, all individuals cross among them absolutely randomly and in absence of microevolutionary forces, okay? On the basis of this ideal situation, the allogamous species evolve through the Hardy-Weinberg law. What does it mean? This very simple law, which is the basis to understand what happens in most nature and allogamous species, is based on the frequency of the two alleles, frequency of the allele 1 and frequency of the allele 2, P and Q, okay? In analogous species, which reproduces and behaves under these ideal factors, the frequencies of the phenotypes can be estimated or correspond to, in the case of the homozygous A1E1, to P squared. I mean, in a population, strictly allogamous population, which follows the Hardy-Weinberg law and its principles, you can use the frequency of the allele 1 to estimate the frequency of the homozygous A1A1 individuals as P squared, the frequency of the allele 1 squared. And the same with the frequency of individuals or genotypes A2A2 as Q squared, or the frequency of the allele 2 squared. And what happens with the heterozygous individuals? The last formula can estimate the frequency of the heterozygous individuals in a Hardy-Weinberg population as 2 by P by Q. And all the three frequencies, if you have did well the estimation, the calculation, would encompass the total 1, okay? And which is the trend in this strictly allogamous ideal species? Well, in just one generation, the population reaches the equilibrium. So, according to the frequency of allele 1 and allele 2, in just one generation of cross-pollination among all the individuals, these both alleles recombine ideally and provide these frequencies, these genotype frequencies of P squared, Q squared, and 2 by P and by Q for the three different genotypes, okay? And reaches the population reaches the equilibrium with this formula. And the population is, by contrast to the autogamous species, which only pure lines or inbred lines homozygous for the gene and two possible genotypes, the homozygous uppercase or the homozygous lowercase. In the case of allogamous species, you have up to 3 gene combinations, 3 genotypes per gene. If you widen this to the different genes that made the genotypes of the individuals, you have a more diverse population than autogamous. Here I have represented very simple, in a very simple way, just considering 3 genes. You can see that there are individuals uppercase, lowercase for A, uppercase B, uppercase, lowercase for C, but also many other combinations in homozygous and heterozygous combinations of A, B, and C genes compared to the autogamous species, which are limited only to 3 gene or genotypic combination, to two genotypic or gene combination per gene. So you can understand that the number of different genotypes in allogamous species, those which follow cross-pollination, are more diverse. And this kind of procedure or reproductive system is better against natural selection. Why? Because they are more diverse population and they are more resilient. They can offer against natural selection more gene, a higher number of gene combinations to face the challenges of natural selection. On the contrary, highly autogamous species have more limited diversity and they have less opportunity to face the natural selection challenges. This is one of the reasons for which in nature, the most common reproductive system is allogamy, is cross-pollination, while only autogamy is common in cultivated crops. Also, in cultivated species, we have a range of allogamous crops, but only in cultivated or domesticated species is possible to find a high number of autogamous species, while in nature, this is not so usual. Okay, here I'm showing you some simulation based on the Hardy-Weinberg law. Again, I repeat, you can estimate the frequency of the different three genotypes for gene A on the basis of the frequency of the alleles A1 and A2, and which is represented on the right is the frequencies of the three genotypes depending on the frequency of each allele. For instance, see in the horizontal scale, when on the left, when P is 0 and Q is 1, I mean, in a population where the allele A1 has disappeared and only allele 2 is present, this is what it means, you can see that there is no genotypes in green, no heterozygous genotypes, because if you multiply 2 by 0 of P and by 1 of Q, it is 0. And also there are no uppercase A individuals, because if P is 0, then the number of individuals in red, uppercase A, is 0. And what happened with the individuals lowercase a? They are the totality. They represent the totality because Q is 1, totality, Q squared is 1. So it is 1, and the rest to the totality of 1 are 0. Lowercase a is 0, and uppercase A is 0. And in other situation where the frequency of the allele A1 may be higher and the frequency of the allele Q or allele 2 is lower, for instance, an intermediate situation, a population where both are 0.5, so half of the alleles present in the population are 1 and half of the alleles of the population are 5.0 Q, then what happens? If we apply the formula, you will estimate that the population has 0.5 individuals of the blue, lowercase, 0.5 of the red, which is P squared, 0.5 by 0, while the heterozygous are the predominant one, which is 2 by 0.5 by 0.5, 2 by 0.25 is 0.5. And in all the cases, the totality of the three genotypes make 1. So these represent different situations with different frequencies of the alleles A1 and A2 and the estimated frequencies of the three genotypes adapted to the Hardy-Weinberg law. I'm leaving you to practice this formula in the case of the frequency of the three genotypes are 0.2 for A1A1, 0.4 for A1A2, and 0.4 for A2A2. And my question are, which are the gene or allele frequencies in these cases? And how will be in the next generation the genotypic and allele frequencies. Somebody could tell me which would be in this population the gene frequency or the frequency of the allele A1 and the allele A2? Roberto, are you brave again? Someone? Well, I'll make very fast. If in this population the three genotypes account for 0.2, 0.4, and 0.44 for the whole value one of the population, means that imagine you have 1,000 individuals, then 200 individuals will be A1, 400 will be A1A2, and 400 will be A2A2. Okay, so the gene frequencies can be estimated directly as frequency of A1, or P is the total frequency of A1, which is 0.2, and half of the frequency of A1A2, which is half of 0.4. Therefore, the frequency of E1, or letter P, here would be 0.4. And what happens with the frequency of the gene or allele A2? The same. The total frequency of the homozygous A2A2, 0.4, and half of the homozygous, 0.2. Therefore, 0.6, and 0.4 and 0.6 of allele 1 and allele 2, or P and Q, makes 1. So we could be in the case of 0.4 for P and 0.6 for Q here in the graphic. And if I leave this population to reproduce according to the Hardy-Weinberg laws, what happens? Watch how will be the frequency of the next generations. I can advance that the frequency of the alleles wouldn't change, but the frequencies of the genes or the genotypes will change, because in the offspring A1 would be P squared, which is 0.4. The square is 0.16. A1A2 would be 2 PQ, which is 2, 0.4, 0.6, which is 0.48. And finally, A2A2 would be Q squared, which is 0.6 squared, which is 0.36. So what we can see is that in this problem, the parent population was not in the Hardy-Weinberg equilibrium, because their genotypic frequencies have changed from one generation to the other. Okay? And we have estimated on the basis of their alleles. And now, after this fast and simple explanation about the different reproductive system, let's explain how the evolutionary forces work with populations. Maybe some of you are familiar to the evolutionary forces. Till now, I have explained you in a very simple way how a strictly autogamous species advance generation after generation, and which is the final trend, or the other extreme, strictly allogamous or cross-pollinated species, how they advance from one generation to the other according to the Hardy-Weinberg law. But in both cases, I have not still introduced the evolutionary forces. Now it's time to introduce. So apart of the own reproductive system, the evolutionary forces play a very important role on the evolution of the populations. There are four, namely natural selection, and in our case also breeder selection, genetic drift, mutation, and migration. Well, how on the whole work each? Well, in the case of natural selection, it works by affecting differently each genotype and modifying their frequencies. And its effect is mathematically estimated through the concept of fitness. Okay, so how works natural selection or breeder selection? Each genotype has a different fitness value from 0 to 1. The genotypes with which better respond, which higher resilience or tolerance to the factors of natural selection, they have more opportunities to pass from their young stage to their reproductive stage. So they have higher opportunities to survive. And it is measured through the concept or the value of fitness. I mean, individuals with a higher response or better response to natural selection, to the factors of selection, they have a fitness close to 1. And that means, numerically, means that these genotypes will have more opportunities to pass from their young stage to their adult reproductive stage, and then more opportunities to have offspring. By contrast, individuals with a lower or worse response to natural selection or not preferred by breeders, their fitness is close to 0. And that means that only a lower amount of these genotypes or individuals will pass from the young stage to the adult stage. And then they will provide to the generation a lower amount of offspring. Okay, so this is how natural selection or breeder selection work through the concept fitness. And on the whole, natural selection decreases diversity. Why? Because natural selection gives more opportunity to some genotypes while decrease opportunities to other genotypes. In such way, some genotypes will become predominant in the population while other will go towards the extinction. So on the whole, natural selection decreases diversity in the population. Genetic drift is other evolutionary force, and it is related to the size of the population. And it is very simple to understand. The lower the number of individuals which make the population, the harder is the effect of random forces. I mean, well, you will see in the practical demonstration, but I mean, imagine that you have the 3 possible genotypes, each with a frequency, okay, and you have a population of 10,000, a total population of 10,000 individuals. By chance, a tsunami appears in the region. Many individuals will die randomly without the effect of natural selection. Okay, the 3 different genotypes will suffer a decrease in their number. But the higher the number of total individuals in the population, this effect, this random effect of a tsunami will affect less to the frequencies of the genotypes. So, okay, the total number of individuals will decrease due to the tsunami. Imagine that half of the population die, okay, from 10,000 to 5,000, but individuals from the 3 genotypes will survive. And more or less, the frequencies of the 3 genotypes will be the same before and after the tsunami, because it's a random effect, a random event. But on the contrary, if you have a small population of, let's be extreme, just 10 individuals, the genotypes with the lower frequency, when a tsunami arrives to the island, if half of the individuals disappear on a population of 10, then this population will become only made of 5 individuals. It is logical to think that those genotypes less represented in the population probably will disappear. So it is a very extreme case, but it's simple to understand the effect of genetic drift. And I will put more detail in the practical demonstration. And now to finish the other two forces, evolutionary forces, mutation. Mutation always increases diversity because mutation provokes the appearance of new genes, new variants of the alleles. So if in a population you have individuals and which result from the combination in pairs of allele A1 or A2, mutation will provoke the occurrence of a third variant. Let's call them A3 alleles. As a result, if this mutation survives in a few generations, new genotypic combination, new different individuals will appear in the population. So it is considered on the whole that mutation provides new diversity, increases on the whole the diversity of the population. And finally, migration refers to migration movements. And on a population on the whole, when individuals leave the population, provoke a decrease in the diversity of this population. But when migration refers to migration with new individuals coming from other populations, it increases the diversity of the population. So migration may decrease or may increase the diversity of the population depending on individuals leave or new individuals with new genes come to the population. Okay, so these are more or less the four evolutionary forces, and now it's time to practice, to show you a practical example. I will connect this. This will be in the presentation. Please limit to practice because maybe the server may collapse if all of you connect. But just take notes because I will ask you to practice. I will use this server now to go in more details about the evolutionary forces, just to show how they provoke changes in the structure of populations. But then it will be time later for you to practice. But please let me practice by myself how this very simple. There are many, but for me, this is probably one of the easiest to understand online simulators of genetic population. It is very simple. Population data. You can see here population data, population finite. So finite, not infinita. We can play with the number of individuals from 10 to 100 to 1,000 to 10,000. We will change, okay? In the allele frequency, I prefer to leave number of population in 5 because in the simulation, the server will offer me 5 simulations, 5 populations, number of generations. I will leave the simulator, the server, to extend the simulation to 500 generations, according to the evolutionary forces, and the frequency of the alleles. This is very important. I will start with a frequency of allele A1 or P of 0.5, which means that the frequency of A2 is also 0.5 as complement to 1. Well, you can see also here fitness. Fitness is the value of adaptation or good or bad response, as I told you, to natural selection, or the response to the selection of the breeder. Okay, 1 is the highest value. So now in the server, the 3 genotypes and A2A2 have the highest fitness value, the highest value. Okay, so they respond very well against selection. Migration, I prefer not to practice to avoid misunderstanding, and by now mutation will be not simulated. Okay, we'll practice just working with genetic drift. Okay, by managing the number of individuals. So I will start just with 100 individuals. which happens and which kind of 5 populations evolve through the generations and according to the P square, Q square, and 2PQ of Hardy-Weinberg laws, how they will evolve. You can see here, okay? 5 populations simulated with the pass of the generations till 400. You can see in the simulation a line in the middle with blue color, which reproduce what happens with an infinite population. If the population is infinite, 1 million individuals or infinite, then the frequency of the allele A1 will stay in 0.5, so no changes. And also for allele A2, 0.5, so the population has no changes if the population is infinite. And this is one of the principles of Hardy-Weinberg laws. But what happens with the other cases related to a population limited to 100 individuals? Some of the population, you can see that the frequency of the allele A1 in this red one has decreased to 0.12 in the 50th generation, then it has increased a bit, then it goes below, it increase, and stay in 1. This means that in the simulation, at the end, after about 180 generations, the allele 1 is the only one, and the allele 2 has disappeared. It has become extinction of allele 2, and any individual which carries allele 2. And therefore, if you make you apply the formula of Hardy-Weinberg, you only will have A1 individuals. 100% of the individuals will be representing the population, and no heterozygous and no homozygous A2 individuals. This happens the same with the green simulated population and with the orange simulated population at the end of 400 generations. On the contrary, other simulated populations simulate that the allele which disappears is A1, and the one who stays 100% is allele A2. Therefore, only individuals A2 are at the end of or after 100, 150 generations. Okay? While the other genotypes have disappeared, extincted. Okay? Well, this is with 100 individuals. And what happens with 10 individuals on the total population? That extinction of A1 or A2 and the genotypes which contain them, which carry them, disappear, but disappear faster. So the lower the number of total individuals in the population, the faster occurs the extinction of one of the alleles, as the example of the tsunami. The lower the population, the more opportunities to disappear some of the genotypes and become predominant the others. On the contrary, if we increase the number of individuals to 1,000, you can see that the populations some are increasing the occurrence of A1, some others are decreasing the presence of A1, but they stay. Okay? Maybe after 500 generations, in some of the population, one of the alleles disappear. But thanks to this high number of individuals, the effect of genetic drift decreases. So I have demonstrated that the larger the number of individuals in the population, the lower the effect of genetic drift, the lower the opportunities to disappear and to extinct some of the strains of the population. Okay? While the lower the number of individuals, the more probable will be the disappearing some of them, and faster in very few generations. So this is the effect of genetic drift. And let's see what happened with selection. I will keep the number 1,000 of individuals. I will keep the frequency of A1 0.5 and the frequency of A2 also 0.5 as logical. In this case, the frequency of the three genotypes will be 0.5, 0.5, and 0.5. Well, let's work with natural selection. Imagine that selection favors A1 individuals, favors but a bit less A1 A2, and goes against A2 because fitness is 0.5 in this case. What happens? You can see in very few generations, in less than 30 generations, the 5 simulated populations have gone to the disappearance of the E2 allele, and as a consequence, the disappearing of A2. Maybe during this process survive because they still have a good fitness in comparison to A1, which are the best. Okay, but at the end, once A2 disappear, even A1 will disappear later, but they will disappear. And at the end, we will have only A1 allele in the population, and A1 population is larger, 10,000 individuals. The effect will be the same, but a bit less acute, okay? Or like this. It happened later. Or let's see, is 0.9 and 0.8. The process is the same, but the A2 disappearance will take much time because thanks to these higher fitness values, they will disappear, but in a slower way. Okay, so this is the impact of fitness or selection. Imagine also that we are breeders now, not natural selection. Now we are breeders, and we can identify A2 phenotypes because they are homozygous and they are recessive. That means that the breeder will can discriminate A2 individuals because they are evident, they are obvious to see, and then they are not left to reproduce. But maybe some of the alleles A2 go with under the heterozygous. What happens? At the end, we will be able to disappear A2 individuals or A1, A2 individuals, but as they go hidden in the heterozygous, the total disappearance of A2 or extinction of A2 will take many generations. This is also the case of rare diseases. As they are so rare, their alleles are very low frequency in the populations, but they go hidden in the heterozygous, and they only appear sometimes when the two parents carry the alleles in heterozygous. Okay, so this is just to practice. I will not work with other evolutionary forces, just with fitness and just with genetic drift. Okay, I will leave sharing this, and I will share with you the presentation. So this is more or less how this PopGen server works, and it helped me to demonstrate a bit more how genetic drift or the natural selection or breeder selection works. So to finish the quiz for you, I will leave you 10 minutes to finish. They are very easy questions. What kind of population is expected in highly autogamous crops? What about the same question in highly autogamous cross-pollinated crops? Which reproductive system provides higher adaptability and resilience to natural selection or to nature? And how selection affects the genetic structure of a population? There are 4 very simple questions. Please send to myself and Ana Marija's email This is important for the certificates. I will put, as always, so I will share, which is easiest for me to revise everything. Okay, the questions of the quiz, what kind of population is expected in highly autogamous crops? Okay, as I explained in the corresponding part, is in a highly autogamous population, it is expected to find a plethora, a mixture of individuals, yes, but highly inbred, plenty of genes in homozygosis and very few genes in heterozygosis. Okay, so for each gene, you may find two different versions, two different genotypes. In our case, I mean the homozygous, but no or very few occurrence of heterozygous genotypes, gene by gene. So this is quite typical of autogamous. On the contrary, on highly allogamous cross-pollinated crops or species, this kind of reproductive system contributes to keep a higher number of diversity, a higher diversity, because it keeps not only the homozygous, but also thanks to the recombination provoked by cross-pollination. For each gene, you can find the three alternatives: homozygous for uppercase, uppercase, or lowercase, and heterozygous, gene by gene. So, allogamous crops or species are more diverse, are expected to be more diverse. A plethora, a mixture of individuals with genes in homozygosis and genes in heterozygosis. Okay. The third question is, which reproductive system provides higher adaptability and resilience? Of course, the reproductive system which provides higher diversity gives more opportunity to this population to face the challenges of natural selection or unexpected events, new pathogens, climate change, etc. And finally, how selection affects genetic structure of a population. As I told you, usually on the whole, general terms, decrease because selection, both natural selection or breeder selection. Selection is in favor of some genotypes and against the others. That means on the whole that the predominant, some genotypes will be predominant generation after generation of breeding or natural selection, while other will go toward extinction, as shown in my simulator. Okay, so these are the answers to the quiz. And just to finish, what we have learned today: diversity equals to resilience, autogamy in breeding, in terms of breeding, in breeding terms, decreases diversity towards inbred lines, provoking also the disappearance of heterozygous individuals or genes, while allogamy, cross-pollination, increases or keeps diversity by recombining genes and alleles. As I told you also in my presentation, these are extreme cases, but there are other intermediate, okay? In such intermediate cases, there are formulae, equations to estimate how the structure of the population advances, considering partly autogamous or partly allogamous, but I prefer to simplify with this, with the unit of today. Natural selection is a very hard evolutionary force and also is helpful in breeding because, for instance, indirectly, when we are acting as breeders, selecting the best tomatoes or the best wheat future population, in parallel, the climate change effects is helping us to shape the population because indirectly we will perceive that there are bad plants, plants which are smaller, plants that did not set well fruits. Probably this is because of the climate change. This is because of nature, which is supporting, which is collaborating with our breeding effort. So natural selection is a powerful evolutionary force and also helpful in breeding. And finally, as homework, please in the next week? Very simple. Practice with the pop gene server, whose link is provided in the presentation. I will copy and paste also in the chat, and practice in the next days. Combine different evolutionary force, mainly the two ones I have discussed with you. And please send 3 screenshots of 3 different data from the size of population and the fitness of the 3 genotypes, and compare and give me your interpretation, your discussion about the results. Compare the 3 screenshots, the 3 simulations, varying, differing the data, size of the population, and fitness of the 3 genotypes, and please interpret. Very simple. Screenshots and a paragraph with the result discussing, and why on this screenshot where I changed the number of individuals, the results are different. This is because, okay, just discuss very briefly in 3 word pages with the screenshot pasted. Okay, I will copy the for you the link, and even you can search in Google as PopGene Radford University. Okay, and you will go directly to this site. Okay, so this is all from my side. Again, if you lose the link as additional material, I can recommend many books on population genetics, but maybe they are too hard. I just recommend at this level or this unit to practice with the link of the PopGene simulator. Okay, so this is all from my side. Thank you very much for your attention, and I will discuss a bit with the next lecturer, with my colleague Michael Snyder, to see something about the next session, Unit 4. Thank you very much for your attention. Hoping you have enjoyed it. At least this simple, as simple as possible approach on population genetics. Thank you.